My goal is just to share an experience that we recently gained by working with Cellecta, and to tell you one story that helped us gain insight in some interesting aspects of tuberculosis susceptibility that we've been studying in my lab for many years. Just a quick overview that my lab studies a specific question is why tuberculosis targets the lungs, and why tuberculosis, what's the mechanism that lead to formation of this necrotic lesions. So we developed a mouse model that is unique in the sense that it develops this human-like tuberculosis necrotic granulomas. This you see bacteria and necrotic cells. And we found the genetic locus that prevents the formation of this necrotic granulomas. So you see congeneric mice that express a resistant locus, they still have mini granulomas, but without necrotization, a big difference. So over many years of research, we found that it's a macrophage-specific phenotype, and now we know that the deficiency in macrophage activation relates to inability to fully activate antioxidant defense. So this macrophages gradually develop this phenotypes that they are damaged by accumulation of peroxidation products, and eventually develop necrotic core of the granuloma. So this is to understand where we are scientifically. But one question that we ignored for many years was why those granulomas in human patients or in our mouse model develop specifically in the lungs, because bacteria disseminates to all organs, but in this particular mutant, we see it in any lymphoid tissue, but it's well controlled. Only in the lungs they develop these necrotic lesions. And we also rejected the hypothesis that it's lack of immunity. You know, in tuberculosis field, people say that lung tissue recruits less active T cells or B cells and cannot really build a nice protective environment. So we immunize mice before infection. We still see the same phenotype, although after some delay. That tells us that immune system can delay protection and vaccine, but it cannot prevent eventually progression and necrotization of the lesions. And to study this in a way that kind of reflects human disease, I don't know how many people study tuberculosis in this room, but mostly human tuberculosis in the lungs that develop in immunocompetent people develops slowly. And it can progress for months or years, or even decades before they form necrotic lesions and even cavities. Many people believe that tuberculosis is lung disease because bacteria directly gets there by aerosol. But in reality, it gets to any organ by multiple routes, but it targets the lungs, and we wanted to make a model that will allow us to address this. So we injected mice in a foot pad, and this is a way to immunize mice. The primary lesion is formed and induces immunization, and only after that bacteria spreads to multiple organs, as we have seen. Now we put preprint that details all the studies, but only in the lungs it eventually progresses. And that's what I want to show here. You can see that in resistant mice, everyone uses Black 6 mice. It's the upper left. You see a small lesion, but in susceptible mice, the SST1 mutant that we study, you see initial lesion with progression and eventually formation of necrotic tuberculosis pneumonia that is typical for human patients. So, and this is a funny experiment I just wanted to mention. In a field, it's one of the hypotheses is that oxygenation of lung is what makes it more attractive for bacteria and allows progression, because tuberculosis grows faster and better in oxygenated environment. But it can also persist in hypoxic environment as well. So here we did an experiment where we implanted lung under the skin of mice and infected them systemically. And after several months, we see that tuberculosis granulomas developed only in the lung implant and formed typical necrotic granuloma with fibrosis and bacteria here. And the spleen implant that was put next to the lung did not develop any visible tuberculosis lesions. So the question, the answer is it's not oxygen, but it's a lung microenvironment that permits progression of tuberculosis in the lung. The problem with this model that we really started to use a lot is that it progresses very slowly and it's not synchronized in different animals because there is some randomness, and mice develop infection maybe after a couple of months or maybe after 6 months, etc. So it's major inconvenience. And then we talked to Cellecta, and they suggested try to use their blood gene expression profiling kit so that we can establish correlation between stage of the disease progression and find biomarkers that would allow us to use it non-invasively and monitor progression of tuberculosis. Every 2 weeks, we collect blood from the mice, and we perform gene expression profiling using this gene-specific transcriptome that Alex presented. And here's the results. So as I showed before, we classify all the mice based on the stage of their pulmonary TB progression, based on their pathomorphology and bacterial loads into 4 stages. And we have early stage, intermediate, and advanced when bacteria start to replicate fast. And then there is a terminal stage that we don't study because you don't need any analysis. You see that the animal is very sick. So what we show here is that when we compared advanced and early lesions, the blood gene expression signature gave us a very clear separation. And now we're doing more experiments where we take blood during the course of infection, plus we treat the mice with antibiotics, and we want to look for the outcomes of treatment using blood signatures. And also when we stop infection, tuberculosis requires long treatment. So if you stop treatment, there is often relapse, and we want to determine whether we can find characteristics that will predict that we can achieve complete treatment, or whether there is a possibility of relapse, so the treatment needs to be continued. So, but one of the specific findings in this blood signature that was not quite obvious from analysis of the whole transcriptome, we found that arginase 1, the marker of alternative macrophage activation, was upregulated in advanced lesions. And then we specifically looked at it. So here you can see that we looked at blood arginase 1 expression in early lesions, intermediate and advanced, and we found that this is just early and intermediate. There is very good correlation between disease progression and determination of this alternative macrophage activation. So more recently, it was not shown here, we've done some module profiling using more advanced analysis, and it shows, like, calculates polarization index, and it shows that with progression of tuberculosis, there is increase in M2-like polarization, but also what's interesting, there is no depression of M1. So oftentimes people think about those like 2 alternative programs, where we found that M1 polarization still persists in the presence of upregulation of advancing M2 polarization. And here what I show in advanced lesions, you have 2 populations, M1 and M2, that colocalize to the same lesion and often closely interact with each other. So this is kind of new biology that we're very excited to study, continue to study, and that was possible to uncover using this technology. Here I just show you arginase 1 staining in early, intermediate, advanced, and terminal lesions. And you can see that with progression of these lesions and formation of micronecrosis and terminal necrotic pneumonia, there is increase in recruitment of arginase 1 positive alternatively activated macrophages. And moreover, now we understand they are not formed in situ, but they are pre-committed and they are recruited from circulating blood. So we are now focusing on these mechanisms. And this is also performed by Cellecta in collaboration, where they looked at spatial transcriptomic analysis of the same lesions. So now we're going inside the lesion, and regions of interest were interrogated for the expression of whole transcriptome using geomics platform. And one of the examples here is arginase 1 and iNOS and interferon gamma, all upregulated in advanced lesions. So the point is that it's not two mutually exclusive states of the cells, but it's a combination of both types of polarized macrophages present in the lesions. So one thing I want to stress that we used 50 to 100 microliters of retroorbital blood drawn from mice, which is we can repeat every week or every 2 weeks for this whole transcriptome analysis. So it's very non-invasive and useful as a diagnostic criteria. But here, in addition, more recently, Cellecta offered to analyze B cell and T cell receptor profiles using methodology that Alex described. And what you can see here is that it's a very good distinction between early lesions and control, non-infected control. And then as disease progresses, there is increase in number of major clonotypes that signify B cell response during tuberculosis progression. And here is my illustration of the fact that this is the lung of black 6 mice that have very small lesions. But these lesions, you can see they have abundant B cell follicles, a lot of in red T cells and iNOS activation, M1-like macrophage activation. This is insusceptible. So very early lesion, very few bacteria, but potent immune response. And we can already see that this activation is reflected in the blood, whether there is not much going on in the tissue. And this is T cell clonotypes as well. So I would say that I less understand what's going on with T cells, but we clearly see that there is evidence of T cell activation in the blood as well. But what I think is the strength of this would be a combination of a transcription profiling of the blood and a B cell receptor and T cell receptor that can clearly tell us about progression and major immune responses that are being activated at various stages of TB progression. So, and the practical application of this would be, besides what we presented here, monitoring disease progression. Now we are working on evaluation of drug responses and prediction of relapses. Then we want to apply this technology to test new vaccines, for example, including subunit vaccines, where we can look at the induction of specific clonotypes and their behavior after subsequent infection and correlation with protection. So with this, I would like to stop and acknowledge my lab members who were able to adopt this technology for the mouse and BSL-3 work. So we have protocols for safe work and containment with all this, and we can share them. And of course, collaboration with Cellecta, Les Kobzik, and Alex Chenchik, who gave us advice and supported through the whole process. Thank you.